Okay, this is for your Chapter 11, Section 1 podcast. Make sure you have the podcast sheet in front of you. We're going to do DNA and structure. So, genetic information. Once Mendel's work was rediscovered in the 1900s, scientists were trying to determine if proteins or DNA was the source of genetic information. In 1952, Alfred Hershey and Martha Chase published results that provided definite evidence that DNA was the transforming factor. Their experiment involved a bacteriophage. Now, what does that mean? It's simply a virus that attacks bacteria. There were two components that made their experiment ideal. The first was the bacteriophage was made of DNA and protein. The second part was that a virus cannot replicate itself. Now, a virus must inject their genetic material into a living cell to produce. So, looking to see if the virus was going to inject DNA or if it was going to inject protein, and that would tell them what the genetic material was. They labeled the protein and labeled the DNA. They then looked to see which one was injected into a living cell. To label it, they used a process called radioactive labeling, which can trace the DNA and the protein. When viruses infect bacteria, they attach to the outside of the bacteria and inject their DNA. They then separated the virus from the infected bacteria. Now, Hershey and Chase tracked the DNA by adding radioactive phosphorus. Now remember, nucleic acids, and DNA stands for deoxyribose nucleic acid. They contain carbon, hydrogen, oxygen, nitrogen, and phosphorus, while proteins have carbon, hydrogen, oxygen, and nitrogen. And that's why Hershey and Chase can track the DNA by adding radioactive phosphorus. Now, viruses later released from the infected bacteria contain the radioactive phosphorus, indicating DNA was the carrier of genetic material. After the Hershey-Chase experiment, scientists were more confident than ever that DNA was the genetic material of living things. This led to the identification of genetic material, but how the nucleotides come together to form DNA molecules and how DNA could communicate information remained unsolved. Now, it took many scientists to work together to determine the structure of DNA. Erwin Chargaff's work led him to conclude nucleotides paired together specifically. Rosalind Franklin, Maurice Wilkins, James Watson, and Francis Crick provided data, analysis, and insights that were pivotal in answering the DNA structure question. Now, Erwin Chargaff came up with a rule in how specific bases pair together. Now, there are four nucleotides, also known as bases, in DNA: A, C, G, and T. That's it. There are no more. There are no less. There are only four bases in DNA. Now, the A stands for adenine, the C stands for cytosine, G stands for guanine, and T stands for thymine. And we call those bases A, C, G, and T. Now, Chargaff found while studying various organisms that the amount of guanine is equal to the amount of cytosine. He also found that the amount of adenine equal the amount of thymine in DNA. So here's how he came up with this rule. Now, Chargaff said that certain bases pair together. Well, if the amount of guanine is equal to the amount of cytosine and the amount of adenine is equal to the amount of thymine, we can figure this out. We know that A pairs with T. We know that C pairs with G. And if C pairs with G, G must pair with C, and we're on the last letter T. If A pairs with T, then we know T pairs with A. This is how we know adenine is equal to the amount of thymine, and that cytosine is equal to the amount of guanine, or guanine is equal to the amount of cytosine, and the amount of adenine is equal to the amount of thymine. And these are the pairings for DNA. A will always pair with T, C will always pair with G. And if you can remember that, then you know that G must pair with C, and T must pair with A. They are complementary bases. Now, Franklin and Wilkins, they joined in on the DNA work too. They joined the faculty of King's College in London, where they fired X-rays at DNA with the staff there. Now, the famous photo is known as Photo 51, and the data collected was used by Watson and Crick later on. We'll take a look at the actual Photo 51 in a moment. Now, Photo 51 indicated that DNA was a double helix or twisted ladder shape. The specific structure was determined later by Watson and Crick when they used Franklin's data and other mathematical data. Now, DNA is the genetic material of all organisms, composed of two complementary strands wound in a double helix. And this is photo 51 right here. And you can see that it's having that double helix, or the double helix strand shape, kind of looking like a ladder. So Watson and Crick were working at Cambridge University in Cambridge, England. Using Chargaff's data and Franklin's data together, they built a model of the double helix and spacing of the bases. Now, some important features of their model include 2 outside strands consist of alternating deoxyribose and phosphate. Cytosine and guanine bases pair together by 3 hydrogen bonds, while adenine and thymine bases pair together by 2 hydrogen bonds. And here they are working together to build an actual model. All right, so in the 1920s, biochemist P.A. Levine determined the basic structure of nucleotides that make up DNA. Now, nucleotides are the subunits of the nucleic acids, and we also call them bases. Now, DNA and RNA share some common traits, but they have a couple unique features. So let's take a look at this table here. So DNA and RNA, they're both nucleic acids. DNA stands for deoxyribose, nucleic acids. RNA stands for ribonucleic acid. The type of sugar in DNA is deoxyribose, while the type of sugar in RNA is ribose. Now, the nucleotides and bases they contain, we already said DNA contains A, C, G, and T, which stands for adenine, cytosine, guanine, and thymine. Now, RNA has A, C, and G just like DNA, but also, instead of T, has the letter U, which stands for adenine, cytosine, guanine, and uracil. DNA is a double strand, has 2 strands, while RNA is a singled strand. So there are some couple unique features that each one has. We're going to be talking more about RNA in the next podcast. So Watson and Crick, DNA is often compared to a twisted ladder. The pairs of bases form the rungs of the ladder. The number of purine bases are equal to the number of pyrimidine bases. So what in the world does that even mean? Well, the purine bases are the pairing of adenine and guanine. Now, we're not saying that adenine pairs with guanine. We're saying the things that adenine pairs with and the things that guanine pairs with are purine bases, while pyrimidine bases deal with cytosine and thymine. So do not confuse this with A pairing with T, C pairing with G, G pairing with C, and T pairing with A. The purine bases deal with adenine pairing with thymine and guanine pairing with cytosine, and the pyrimidine bases are cytosine pairing with guanine and thymine pairing with adenine. Do not confuse those two. Now, complementary base pairing is used to describe the precise pairing or purine and pyrimidine bases. It is the characteristic of DNA replication that the parent strand can determine the sequence of the new strand. And we talked a little bit about this in class, and we're going to even talk more about it in the next class. So if you guys have any questions, please make sure you leave a comment below or bring your questions to class. We'll see you guys next time.